Brilliant. So welcome everybody to this webinar. My name is Professor Salman Siddiqui. I'm a respiratory professor of experimental medicine at Imperial College in London. And I'm really delighted to host this BioReam network webinar today. And the provenance to this webinar is that naturally there's been an expansion of mathematical and computational modeling in the respiratory domain area. And we wanted to really explore this from the perspective of getting it into the clinic, democratizing these models, very much like ChatGPT, which is everywhere now, even in the clinic, basically, and in the clinical domain for clinical doctors and healthcare professionals. And we've got a fantastic, amazing lineup of speakers, actually. I'm really delighted that they've been able to take the time and join us today. And so we're going to start off today with Professor Peter Hunter. I'm just going to give you a brief introduction to Professor Hunter. He's a distinguished professor in bioengineering and the founding director of the Auckland Bioengineering Institute. He's also the co-director of Computational Physiology at Oxford University. He's been a real pioneer in this field and established the Physiome Project of the International Union of Physiological Sciences. And he's going to really give you a tour de force and insight into standardizing tools for modeling multi-scale physiological systems. Peter, thank you very much, and it's over to you.
Yeah, thanks, Salman. I'll just share my screen. And Ola, can you see that okay?
Yep. Yeah, we can see it.
Okay, so I'm going to talk about standards and tools for modeling multi-scale systems. And I'm going to talk about four particular topics. One is just a little bit of background in terms of the International Union of Physiological Sciences Physiome Project, which is how we started a lot of this, and how that's led from CellML to what we call the Physiome Journal. Then I'm going to talk, the next two topics are really the main focus of what we're doing now, which is building scaffolds and mapping a lot of neural and vascular, etc. systems. But also I want to talk a little bit about how we've come to see the need for a very solid physics framework for multi-scale modeling, which we've never really had, I don't think. And then I'll talk a little bit about future work in terms of the virtual human, and Lies will very much carry on that in that area. So first of all, the background. It really started with Dennis Noble, Jim Basingthwaite, Marco Visconti, and Stig Omholt. And this was about really trying to get people together around developing tools and standards for physiological modeling. It also, and that was in around 1997, but it also kicked off later discussions in the US with the NIH IMAG program led by Grace Ping, and in the EU, the Ilias Ikiavadis, shown on the left there, which was really the beginning of a lot of the framework 6 and then 7, leading now, of course, to the Horizon Europe investment in virtual humans. But that a lot of that started with particularly Ilias Ikiavadis's efforts to get modeling onto the agenda for the European Commission. And then, so what we really focused on initially was the development of standards. CellML, I'll talk more about this in a second. CellML and a Physiome model repository, and then open source, freely available open source tools which would implement those standards, particularly OpenCore. And then more recently, it's led to the development of a journal, which I'll talk about. So the idea behind the standards was just because biology is so complex, because the mathematics get so complex, we need to be very, very careful about being able to be reproducible. So a lot of the effort in the early days was just to get in place standards that would allow models to be put into a form that could be demonstrably reproducible, so somebody other than the author of the model could make use of it. And we developed the standard and a website and a repository, which currently has about 1,500 models covering a wide range of physiological processes. Really all of biology, primarily to do with lumped parameter models, but also including some spatially varying models. And then we decided a few years ago that we really needed to provide more incentive for people to put the effort in to make their models reproducible. And so we developed the Physiome Journal, which is we think of as a secondary journal. So you publish your primary paper in a normal journal, but then you publish an associated paper where you put the extra effort in to make that model reproducible and obey FAIR principles. And so it's got a very low, it's open access, very low access author charge, and it's still really just getting off the ground and going, but there's about 20 papers there now which you can look at, which demonstrate what we're trying to do in terms of making these models, putting the effort in around curation, documentation, etc., around the models. Then the next thing I want to talk about is this concept of scaffolds and mapping. And this has very much been driven by an NIH Spark project. And what this is about is we've developed a computer-aided design package really for anatomy, again, open source, available on GitHub, and all the tools and documentation is at that website there. And this is about, it's been led by Richard Christie in particular and Hugh Sorby. This is about being able to design complex shapes for carrying spatial information in biology. And those shapes could be relevant to a small piece of tissue, or it could be a whole organ, or it could be the whole body. So it's really a CAD package for being able to develop suitable spatial frameworks for handling information. Spatial information. And it's been driven by, we're into the seventh year actually, of our NIH funded Spark project. And this is about the autonomic nervous system, and where we're mapping a lot of the neural pathways for multiple species, but in a highly curated, with all the provenance given for all the statements, and working with groups, for 3 other groups that are part of an effort to support the whole autonomic mapping and modeling frameworks. And what we're doing with the scaffolds here is just basically designing spatial scaffolds that allow data to be embedded in a coordinate system that provides a reference for material coordinates within those scaffolds. So the scaffolds are built up from components using that CAD approach that I mentioned earlier, and that can be, for example, building a whole colon from defining the center line, defining the haustra and the coli, and then being able to build a finite element description of the anatomy into which you can then embed all sorts of protein or genomics or histology data, and being able to develop those scaffolds so that they're applicable across various species, in this case, human, pig, and mouse. These are scaffolds for the heart and the lungs. And in all cases, the scaffolds are designed to provide that definition of a coordinate system, common framework for mapping data, but be able to export in high resolution meshes for computational work. And so this shows some of the mapping of autonomic system data into the atria of the heart. This shows mapping data into onto the stomach. And one of the big advantages of this common coordinate system is that you can then map data from multiple groups. So here's data about cell type and the density of various cell types in the stomach coming from a group in Melbourne, John Furness, but also a group, Terry Powley at Purdue, mapping the vascular system, and Jack Chen in Florida mapping the autonomic system. You can bring all the data sets into one common framework that's where the scaffolds can be adjusted to fit the experimental preparation that's being used to collect that data. So you're pulling all the data into one common framework for being able to use those data as part of a computational model. And then we embed those organ scaffolds into whole body frameworks. So then the other major focus for us at the moment is really trying to understand how to deal with multi-scale physics in a much more consistent way than in the past. A lot of the lot of the development of standards have enabled reproducibility and demonstrable adherence to fair principles and showing the reproducibility of a model, but it doesn't mean the model is correct. The model could still be based on incorrect physics and be reproducible, but not that much use if you don't understand the limitations of the underlying physics. So we really went back to the drawing board and started thinking about what the appropriate framework for dealing with physics in a multi-scale sense, and hence the use of bond graphs, which are a way of just guaranteeing that you always satisfy mass, charge, and energy for all your systems, and you can then deal with physiological systems that move energy about between mechanics, electromagnetics, and chemistry. And the bond graph framework is proving very valuable for making sure that we can capture behavior at the protein level, but also at the whole body level, which I'll talk briefly about. So just as an example, if you look at one particular part of the genome, the set of SLC, solute link carriers, these are the transporters in membranes, all cell membranes that transport small molecules across those cell membranes. And there's about 66 gene families, and that represents about 2.5% of the genome, 458 genes currently. And these transporters are not ATPase pumps, that's a different class, but everything else basically that isn't using ATP or isn't an ion channel. And these transporters are responsible for glucose transport, sodium, calcium, everything you can think of in terms of non-pump based transporters, pH control, for example. And if you look at the various types of chemistry going on in these transporters, they're typically either facilitated diffusion of one molecule through that in either direction through the membrane, or they're co-transport or antiporters, often using the sodium gradient to assist the delivery of, say, glucose into the cell. Or in some cases, they involve 3 ligands, and in very few cases, 4 ligands in both directions. And what we're doing is saying, can we represent the physics at the scale that's of relevance to physiology? Can we capture the physics that make sure we're obeying mass, charge, energy transfer in templates that we can then parameterize appropriately for any of those 458 proteins in those gene families? And I think the answer to that is yes. We're not quite sure of how many templates we need yet, but here's just an example. So if you look at glucose transport, which is carried by 2 types of SLC transporter, GLUT2 protein, which is SLC2A2, and SGLT1, which is a sodium-assisted glucose transport. And so the physics here is just glucose entering by binding to an enzyme that's rotating through a number of different states. Binding is very fast, unbinding is very fast, and you represent the reactions for the binding and the transition of that protein from outward facing to inward facing as it delivers glucose to the inside of the cell and then transforms back again to pick up the next. And likewise for SLC5A, it's a sodium assisted, so you're using the sodium gradient to help drive glucose into the cell. And it's a slightly more complex pump, and because sodium is being transferred, it's electrogenic, which means you've got to deal with charge transfer as well as the molar flux of the ions. But it's very straightforward to develop models that we've shown that they can very accurately reproduce the experimental data on the behavior of these proteins. And very often you can derive analytic expressions out of the bond graph models that tell you exactly what the flux is under steady state conditions, when you can assume that particularly the solutes are present in a much higher concentration than the enzymes in the membrane themselves. So you can get much faster computation by deriving analytic expressions for the solute fluxes. So we tried to do that for a wide range of these transporters. Okay, and then the last thing I want to talk about is just the process of using that multi-scale framework for virtual humans. And I think, really this is about taking the Physiome project into the clinic and personalizing it much more. So very much the ideas that are now being driven through the European Commission and the EDIS project that Lies will talk about, but also a lot of interest, I think, in the US, increasing levels of interest in the US in having a virtual physiome or virtual human project. So for me, it's really about the Physiome project, but applied in a way that you can personalize models and with a strong clinical focus, and you can make them more generic than personal, but often it is associated with that concept of a twin. So here's the basic. This is our 12 Labours project in New Zealand, where we're funded by the New Zealand government for a project that's looking at multi-scale modelling as part of developing these whole body models focused on the lungs, the heart, the musculoskeletal system, and particularly with the autonomic system, the stomach, and the uterus as well for women's health issues. And what these models do is to use this framework from cell level behavior modeled with bond graphs through developing, and I'll talk a bit more about this in a second, functional tissue units up to organ scale models, which use often finite element descriptions, and then surrogate modeling to reduce the complexity of these to go into bond graph models at the whole systems level. And what this is trying to do is to have the ability to link data from across different levels, from blood biomarkers through to clinical images for MR, etc., through to physiological function tests, up to in order to integrate the data sets that are associated with making a diagnosis for a given individual. I mean, it's very, very early days for this, but I think we are getting in place a framework to handle this multi-scale side of it. And it's also strongly linked with measurement. So the ability to have both implantable, wearable, or clinical imaging devices, for example, that allow the models to be coupled into measurable parameters from a whole range of devices. So the 12 Labours project is very much building the multi-scale framework, but also looking at how it couples into devices and thinking about electronic health records and how we couple into the health system much more. And so that means we've got to think a lot about systems physiology, homeostasis at the whole body level, where bond graphs can deal with systems that are distributed across the body, and then link them down to tissue level exchange, where we've got to be able to deal with individual proteins within functional cell units and link them to exchange processes for linking into whole body physiology. So we've developed this concept of a functional connectivity map, FC map, which is on the Spark portal and has all the organ systems defined, all the organs, and then all the functional tissue units within each organ. And it's a street map-like environment where we can, all the information, which includes all the autonomic connectivity and vascular connectivity, has provenance. You can look at the sources of information for all the connectivity. So this is all on the Spark portal. And the idea is that we have with the FC map, the idea is that we have a single knowledge base which we can interrogate with a Google Street Map like view. So we can zoom in, we can see the big picture, and we can decide what elements we want from the big picture, but we can zoom into the detail as well. And here's an example for the kidney going into the proximal convoluted tubule, looking at the functional tissue unit at that scale, going down to the level of an intracellular unit, in this case glucose control using gluten SGLT, and then down to these individual transporters that I was talking about earlier. So the framework, I think it's really important to get a very robust, properly curated, with all the provenance there for the information, but where we can link across scales, but across organ systems, I think is the only way we're really going to handle some of the challenges that we face with whole body modeling. So I'll stop there. Thank you.
Peter, thank you very much for a wonderful talk, actually. And I propose that we save questions for the Q&A at the end. So hold your thoughts for Peter. So we're going to come to a Q&A at the end. I'm going to now move on to Professor Liesbet Geris. I'm just going to introduce Liesbet, who I've not met before. It's lovely to meet you this morning, Liesbet. Liesbet is a professor in biomechanics and computational tissue engineering at the University of Liège and KU Leuven in Belgium. And she's the scientific lead for the Prometheus platform for skeletal tissue engineering. She's also the executive director of the Virtual Physiological Human Institute, and she's the lead for the European Commission-funded consortium EDITH, which is all around developing a new ecosystem for virtual computational digital twins. And she's going to tell us more about that. So over to you, Liesbet, and thank you for agreeing to talk today.
Thank you very much for having me. I'm looking for the right window to share. There we go. Put it in full screen.
Brilliant. Yeah, we can see it now. Yeah.
Awesome. Okay, so thank you very much for having me. It's a pleasure to be with you digitally this morning. Peter has already indicated several times or referred to the EDIT project. So I'm happy to explain you a little bit more what this is. EDIT stands for Ecosystem of Digital Twins in Healthcare, and it's a coordination and support action funded by the European Commission under the Digital Europe program. Coordination and support action means that we are not there to do research, but that we're there to bring the community together and build or propose a roadmap how to realize a vision. And in this case, it's the vision of the virtual human twin that I will explain in detail a bit later. I will first explain, if I try to get rid of this beam, there we go. So first explain maybe the concept of digital twins, not so much the concept, but the definition that we're using, because there is some controversy on the exact wording of that definition. So digital twins as a concept, when it comes from Industry 4.0, is a virtual representation of a physical object. But what is more important is that it requires a bidirectional, real-time, and dynamic interaction between both. So reading out data from the physical twin, feeding it into the digital twin, and then being able to dynamically alter or give feedback to the physical twin again. Now in healthcare, this real-time reading out of data is not always possible, or at least not the data that we need. And also dynamically recalibrating doesn't really make much sense in most healthcare situations. However, some people feel very strongly, so we come to a definition that looks a bit more like individualized computer modeling. So using individual specific models for all the elements of the entire healthcare system. Now, some people feel very strongly, though, that you cannot use the word digital twin without having this feedback between the physical and the digital twin. And recently, the National Academies of Science, Engineering, and Medicine in the US did a whole study on digital twins, and they came up with a very nice, I would say, Belgian compromise between those that say it doesn't make sense and those that say we really need to have it in order to call it a digital twin, and that is to put the human in the loop. So the feedback from the virtual representation to the physical counterpart can happen through a human-in-the-loop decision-making process. So this actually unites both definitions. So when we talk about digital twins, this is what we're talking about. Now, technologies for digital twins, as I'll be talking about a vision and a platform and whatnot. So not so much about different technologies that we'll use. I just want to make the remark that when we talk about digital twins, we do not make any assumption on the technology that we are that is used to build the digital twin. So it can go from the black box data-driven tools like AI, machine learning, to the white box models where you actually know, you take the knowledge of physics and physiology and you put it into the model, down to what is most often used. We use what we know, and then we supplement with data to cover the things that we are not really sure of. Oh, and all of this is at the moment, but I'm probably preaching for the converted. This has made it into healthcare in various ways. For instance, as a fourth pillar of evidence that is accepted by FDA and by other regulatory agencies to establish information on safety and effectiveness of therapies. So after animal and bench testing and clinical trials, there's computer modeling and simulation. And this is used not just in the design phase, where you have here on the left in the loop, the optimization of the design through virtual prototyping and testing. It is also used in preclinical and clinical elements where already 5 years ago, there was an adaptation of a pacemaker approved by a full in silico clinical trial without going to a human trial. And then, of course, all of the data that we get from the post-market monitoring phase can be used to further optimize products going forward. Now, what we have been doing inside the community to prepare is to try and understand what it is that clinicians in general know about in silico medicine, because of course, you are here. That means that you are interested. We all have in our projects people that we are talking to that are very well aware of what it can do and what they might expect. But we wanted to have a sort of a feel of what is more widely known about modeling and simulation. So we did a survey within the clinical community that was driven by Claudio Capelli from Gauche and UCL. And we really wanted to map what are the needs and the fears and the expectations. From this, we did an analysis and a SWOT analysis, and you see the word trust coming back in multiple different areas, because this is really what it centers around. There is already trust that this will be able to have a positive impact, and they see it as an opportunity to add additional trust in the decisions that are made. But it's also a weakness because they don't know how technologies work, what is really necessary in order to do this, and as a threat because maybe putting your trust in a tool like that might be problematic, or there might be over expectations. So we are currently running the survey again to see if in the 2 years since the first survey there has been a lot of change in the attitude towards in silico modeling. And then a final bit before I go to Edith is the good simulation practice, because part of trust, of course, is seeing how it works in your hands, but also having some objective measure of credibility of the models. Peter already referred to reproducibility and mentioning that because it was reproducible might not make it usable. You here have an additional proof or an additional element to show credibility, and that is doing verification and validation. And as a community, we wanted to make sure that this would lift, like say, the bar for the entire, that we would increase the quality across the entire ecosystem. And then through that, we had, with Marco Vicciconti and help of the Avicenna Alliance, we had a grassroots movement of people working together in the community of practice in Slack, working on what we call a good simulation practice. Of course, we cannot define a good simulation practice. So the resulting white paper that actually turned into a book is called Toward a Good Simulation Practice, where we lay out, following the good clinical practice, what are the different elements that are required in order to build credible models for a healthcare setting. And FDA has been very active in reviewing all of the chapters. And so this recently came out as a free ebook. Now with that, so this is where we are in the field. And Peter already explained the 12 Labors project and all of the work that the awesome work that they're doing in Auckland. The European Commission has discovered that they have made a lot of investments in digital health over the past decades, but that these things are not always maintained, that sometimes people are reinventing the wheel and that there's a lot of fragmentation in the landscape. So they wanted us to think about how to come up with a platform or an infrastructure that would lead ultimately to the virtual human twin, so the integrated virtual human twin, and would make sure that the investments that they did and all the work that was done would not get lost going forward. Of course, this is not starting from nowhere. There are internationally and within Europe a lot of good examples where people are integrating models across scales, models across organ systems in order to answer questions. A lot of the work that we currently see that has made its way into the clinics or has made its way into industry is focusing on single scales or on single organ systems. But in terms of research, we have already taken a few steps further, obviously. The thing there is, though, a lot of times when people are making these multi-scale models, there is a lot of reinventing the wheels, or people are not entirely sure how to reuse things that other people have developed. So this is why we came up with the definition of the virtual human twin, which for us should be a systematic and ever-growing digital quantitative representation of the actionable knowledge available on human pathophysiology. It is to be a European platform that will enable the pooling of resources and assets to develop these digital twins in healthcare, meaning resources are data, models, algorithms, but also access to the compute power infrastructure, to the storage networks. So building these digital twins and assessing their credibility. And so this federated public infrastructure should be able to link up to existing other infrastructures and allow for this collection of appropriate resources that have not yet found a home. So the data that is not yet in another repository should find a home in the VHT infrastructure. And all of this, of course, requires the engagement of a collaborative ecosystem. So what does that mean in practice? That means that it should be an interface where you can go from your own computer, you can search the catalog by data type, anatomical location, age, whatever attribute that you are interested in, and find data resources. Now, in some cases, the data or the resources might be available in the infrastructure. In some cases, it will lead you to catalogs of other organizations or hospitals, or ultimately the European health data space. So it's not necessarily because you can see it in the catalog that you can access it. But in any case, you should be able to find it. You should be able to access every digital twin that was developed in Europe, funded by European money, including your own. And then you should be able to use, or the platform should offer possibilities to find matching data or matching resources. So if you have a model, you're interested in the data, using the right taxonomy, the right metadata, you should be able to find matching data or the matching model, depending on what it is that you're looking for. The infrastructure should help you run all these models on your own HPC system or HPC centers that have expressed the interest in running a specific type of application, or apply for resources to be able to run it somewhere. Then it should also help you orchestrate multiple digital twins, so build multi-scale or multi-organ systems, and then script the whole thing. So help you develop workflows that will allow you to redo this work, or use somebody else's workflow that should also be available as a resource in the platform, so that you can more easily run what it is that you want to run. And ultimately, you should be able to take out, this is a public platform, you should be able to take out all of the tools that you've used with the appropriate acknowledgements and interactions with the people that put tools there, and build a standalone application that then can make its way into applications such as a clinical workflow or into a business. Now, in order to realize this, and I realize it sounds quite naive, this is where we want to evolve towards. But in order to do that, there's quite some work that needs to be done. Of course, on the technology side, on the individual resources, because there's never enough data or never the right enough of the right quality and type of data. The integration of the resources, if you want to build multi-scale models, how do we do that? And Peter's solution is really a great example of how this can be done in a way that is reproducible across organs and across scales. The right APIs, the infrastructure, and connection with things that already exist. But it's not just the technology part, obviously, that needs to be handled. There's also a whole ELSI part, the ethical, legal, social issues that need to be addressed. So access, the ethics, how do you deal with this data, the legal and policy aspects, because there's a lot of policy that is being developed now with the AI Act, the Health Data Act, or the Data Act. There is the EHDS that is coming. There are a lot of regulatory considerations. If we want to ultimately take this to a patient, then this needs to pass regulatory approval. how can we already help build this in? For instance, FDA has launched all their regulatory science tools. Can they become part of such an infrastructure that will help users by including these regulatory science tools early on to facilitate regulatory approval later on? And of course, standardization, also mentioned by Peter, is an important part of this. And for some parts, this has really been done. There are the CellML and other standards. But for some parts, some technologies, some in silico technologies, this doesn't exist just yet. So there's work to be done. There's then also the different users that might have different requirements, different expectations, different levels of knowledge by which that they have when they access the infrastructure. And you might want to change the way they interact with the platform based on that. Like as a starting PhD student, I want to see everything that is in there. Whereas if maybe I'm a clinical user, I don't want to see all of the very early research models and developments. I want to see the things that are already having a certain level of credibility, so that have already passed certain steps of scrutiny. And I want to be able to interact. I don't want to have to go to the VHT and then have 20 other user accounts for different other platforms. It should be a one-stop shop, so that and that helps you access the other platforms and repositories that are available. And then, of course, there's a sustainability that needs to be worked on. How can we get this into clinical workflows? How can we interact with large companies and startups? How can we build businesses on those? And how can we sustain this as a European infrastructure? So very briefly on implementing the vision, because the coordination and support action does not need to implement this. We need to write the roadmap on how this will be done and how we foresee this as a 10-year program that will take, of course, much more public funding to realize. We do need to provide a proof of concept repository and catalog. And what we are working on is a very standard layout of this repository. There is, if you look at the bottom right in the greenish part, there is the part for providers. If I want to upload something, what is the taxonomy that I need to use? What is the information that I need to provide? How is this assessed? What are the standards, the requirements? So all of this is being tested. We have the admin part, but of course the identity access management, the curation, the validation tools. And again, Peter mentioned already quite a lot of this. And then there is in yellow the catalog part with the resources for the users. How can they access this? How can they find this? Then you see here in the dashed green line the link to the simulation platform, where we have a very high-level proof of concept implementation, just to show how it might work with a very classical front-end services, middleware, and link to the infrastructure as a service and the HPC layers. This is modeled based on, for instance, the eBrains platform and a number of other platforms that exist. And there recently, just last week, the Digital Europe program launched a procurement for the building of this, the actual construction of this simulation platform, for which they've foreseen initially 24 million over the coming 4 years. Now, in order to really have a good idea of what is expected and what might work, we have a number of pre-selected use cases in the EBIT consortium that, as you can see, cover different diseases, different organ systems, different modeling types, so that we have a wide variety. We have different data requirements, different compute requirements, different ways in which they can integrate with one another or with pre-existing platforms, and then different users or different end users for these models. In the meantime, we've also opened the proof of concept infrastructure for other external use cases, and people have been submitting proposals or their own use cases, their own resources for inclusion into this repository. And this helps us to make sure that what is written down in the roadmap, in the vision, actually is covering all of these different things. We've been talking a lot to industry, large industry, small industry, medtech, pharma, and software, to understand how they see their possible interaction with such a platform. And of course, their value proposition depends on who they are and who you talk to within the organization. But roughly, it comes down to seeing interest in using the resources, using the publicly available portion as a sort of sandbox to test their new developments, use it as a benchmark for their own developments. And then everything that is done in terms of technology development, in terms of establishing ethical, legal, social clarity and certainty, like liability, IP, all of this, when it's sorted for a public infrastructure, it will also provide more clarity and certainty for them so they can use these developments. And then, of course, as a networking tool to find other partners and to find other commercial opportunities. We've also been talking to clinicians on what it is, how they see the interaction. And then, of course, it depends on who you're talking to, whether you're talking to researchers, clinicians in training, practitioners. There, it really depends on how much time they can invest and what it is that they want, what the shape will have to be of this VHT interaction. So in training, it might be training on virtual patients, testing hypotheses, looking at comorbidities. But as a practitioner, once it has to have regulatory approval and it needs to find a way into a clinical workflow so that it facilitates the life of clinicians rather than adding yet another thing to it. All of this we have put together in a first draft of this vision and roadmap that was submitted last year. That was a 100-page plus document that listed, it's not so reader friendly, but it gives a good overview of what we feel are essential elements that need to be addressed in the final roadmap. We've gone through a process of, we established this as a consortium with experts, and then we've gone through several public meetings. There was a public feedback phase that led to this first draft, and that has been downloaded now nearly 2,000 times. So people are really engaging with it. And now we are working on the follow-up, so the final draft or the final version that needs to be ready before the end of this year. We're also working on the sustainability and the ecosystem involvement, so to make sure that we make policy recommendations on how the policies that are currently being discussed in the EU should facilitate or could facilitate, and how what we need to be careful for, because in some cases the policies that are there might make it more difficult for this vision of the VHT to be realized. Of course, we are working on promoting the buy-in, because this is not something that can sustain by just European top-down funding. We are working on a national EU27 strategy, member state strategy, so that there is also bottom-up funding for this. And then develop incentive mechanisms for researchers to understand why they should be able, what it is advantages for them to contribute to this. And then there are all kinds of mechanisms, like also the Physiome journal that Peter mentioned, because if you do this, you get something out of it. So, but just last 2 minutes, if you would want to contribute in some way, shape, or form, here are some options for you. As I mentioned, we have the clinical survey open. And so if you're a clinician, you're very welcome to share your thoughts in the survey. It will take you 10 minutes to fill out, and it would provide us very useful information on the current status in the clinical field. We noticed that sometimes we hit on a network, and so now, for instance, we have a lot of German anesthesiologists that filled up the survey. So we're trying to get this balanced out by other disciplines and other countries. We have use cases. If you have a data set lying around that is not personalized, or that is more anonymized, or from an in silico study, feel free to contribute this. There is a manifesto. We have European elections coming up, and as we all know how these things go, there might be some shifts. So we have written a 2-page version of the VHT vision and recommendations, and we've gotten this to sign. We've called it the manifesto. We've gotten this to sign by large companies, European ones, but also globally, universities, hospitals, patient organizations, clinical organizations, in order to show the policymakers that this is something that people are interested in and want to see more investments in, to help us to provide more leverage also to our colleagues within the Commission. And then we're of course writing the roadmap. We've recently reopened it, so there's still a draft on Zenodo, but there's also per chapter a Google Doc that you can actually go and read and contribute to. This been distributed in different chapters, so you don't have to read everything to go and to the place where you are interested in and contribute. And then we have also a clinical working group where we try to work on incentivization and requirements and user needs to make sure that this clinical perspective is very well represented within the roadmap. And then finally, we have our final ecosystem meeting for the entire ecosystem in July 15 and 16 in Amsterdam. It's a free meeting. You'll be provided with lots of good food. You just have to get yourself there. And there is also support for this if you want to. We have keynotes from the Commission, from FDA, from clinicians, from various members of the ecosystem. And we intend to make this a very interactive meeting, which should go towards validation of the roadmap and a consensus on the recommendations that we want to make to the European Commission to further support this whole initiative. And with that, I'd like to thank you for your attention.
Thank you, Liesbet, for a terrific talk. Actually, I really enjoyed that. So we're now going to open this up for the Q&A, and to facilitate this, I've got my co-chair, Carl Whitfield, who's going to start with an opening question. Carl.
Yeah, thank you very much, Liesbet and Peter. They were great talks. I suppose I'd like to ask about it. If you have a sort of, I mean, you both touched on this, but if you have an idea of how, like almost a worked example of how you envision in the future, something like this might work in the clinic. Like, am I imagining a sort of integrated system that the sort of clinician doesn't really see, or are we thinking more like clinicians being more interactive with these platforms that you're talking about?
Peter, you want to
I can.
Yeah.
Okay, very briefly. I think the way that we're primarily thinking at the moment in terms of that clinical linkage is, or how we produce a workflow that fits into the clinical system, is to essentially produce the same sort of reports that the current that are in the electronic health record system, but produce them in exactly the same format from the modeling, from the predictive modeling, so that the clinician is actually looking at a report that is exactly what they're expecting to see given a particular condition. And so we're trying to make our outputs tie in as much as possible with the way that the clinicians currently look at information from reports and from measurements. But it's I mean, I think it's very early days for this. A lot of the implementation of these models into the clinic so far has been around single organs or particularly single levels. And there's no great examples so far of really dealing with the multi-scale challenge in the clinical setting.
And I think on top of this, an analogy that I heard that I thought was interesting was the way that other technical technologies made it into the clinics. Imaging, everything that had to do with imaging in the very beginning was in a centralized imaging department, where all people had to go there for all kinds of imaging. And then as technology became more easy to use, people had an increased understanding and an increased training also from the base training. You see that there's multiple imaging technologies that are now in the different departments already. But then for the more complicated ones, you still go to the radiology department. So modeling and simulation could also be something like this, where for individual smaller tools that can have outputs that really can be included into the clinical workflow, they become decentralized. But there is also a more centralized service where you go for more for those types of modeling, where you actually maybe can do less automation or where more expert input is still required. So it could also be a bit that strategy that could be followed initially.
It's a wonderful response. And Liesbet and Peter, if I could follow up with a question around ownership and democratization of the kind of models that you've talked about. Because ultimately, clinicians, as you say, are going to use these models when they understand them, when they feel part of the community, when they own them. I want to put it to you, statistical models embedded in frameworks such as R are now commonly used by medical students. Medical students are now learning Python as part of their first year undergraduate curriculum. But I don't know about in the Netherlands or in Auckland, but in the UK, you don't actually have to even do maths and physics to become a doctor. So I just want to put that back to you. You know, many of the kind of techniques that you referred to do require quite a high level understanding of physics and maths. I just want to give you a very open question. Given that the end users of this technology will be doctors, and doctors will never use this in any way unless they can open the bonnet and at least have a cursory understanding of the techniques, as they do in art, you know, as a good example. What do you think is needed to create that real shift in the community so that the medical community really embrace these multi-scale and computational models, as they have done with biostatistical models, R being a good example?
I can tell you how it's worked best for us in Auckland, and that is where we have medical students who then go on to do PhDs within the ABI, within the Bioengineering Institute, and then go on to clinical practice or starting up companies. That often is the best avenue for getting these technologies into clinical practice, is via these people that have trained within the ABI and then are going into either clinical practice or into companies that then promote the use of those technologies for clinicians.
Thanks, Peter. So here are calls for incubators for people that are minded to approach these areas, Liesbet.
And I think it's not, you need to have some understanding of or trust in the technologies that go below, or that go into the development of these twins. But that doesn't mean that you need to be able to necessarily execute all the models yourself. Sometimes it's enough to also have an understanding of how to assess credibility of something. So what is the evidence that I want to see? What are the checks that I need to do that I want to see before I will put my faith in something? Like, I don't need to know how PCR works, but I need to know that if I look at a PCR curve, that it needs to be within 30 cycles, or else I shouldn't be trusting it. So there are a number of different ways or different depths in which you need to know and be able to do something before you can trust it. And I think that's also part of the education. So this credibility assessment, if we can make sure that this becomes also within the compute community, if we start using these terms, and if we start diligently always showing credibility assessment in our models, then the quality increases, and it becomes also much easier to explain in more standardized terms how to look at models and how to when or not to trust outputs of models.
And of course, the same happens in a number of other disciplines. I mean, in engineering, engineers use element packages without necessarily understanding exactly how they work. But they as Liesbet says, I want to make sure that it's giving them results that are sensible in terms of their understanding. But that's common across all professions. You learn to use tools where you don't necessarily know a lot about the physics underlying that tool.
Thanks, Peter. So I'm going to hand over to Peter Robbins, who has a question. Peter. Peter, you're muted just at the moment, so we can't hear you. Peter, you just need to unmute yourself. Just to unmute. Let's try that. Oh, perfect.
Thank you. I'm very interested in this interaction between the digital twin and the physical reality, and you can imagine that being a patient and the patient having their digital twin almost. And I am quite interested in how you actually go about taking observations from the physical side of it and putting it into a patient model. So, for example, it's very easy if you go down the scale chain, and what you've got to put in is the equilibrium constant for the particular isoform of whatever pump or something like that you've got there, and you can put in bespoke coefficients into that, and everything then bubbles up through the model because you've changed it at the bottom end. But if you were doing something like, if you take an intermediate thing like measuring a hemoglobin concentration, some models that are around, you just put the hemoglobin concentration in and you could have that as part of your transport system. But if you've got a model where the hemoglobin is really something that comes out as a dependent variable, say you've got the iron uptake and so on model, and you've got the EPO modeling going on there, then it's very difficult to just take the measured hemoglobin and put it into the model. And then if you move further along the chain of physiological measurements that you can make, say you do something that's pretty integrative, that's done. A good example of that would be a incremental exercise test or something like that, where you do it and you're looking at, you know, what happened. Well, you'd be looking at sort of the cardiac things, but you're also very interested in sort of what is their maximum oxygen uptake and so on, those kinds of things. And those depend on all sorts of things. And I don't actually work, what I struggle with at the higher end of the measurements from patients is how you actually take relatively integrated measurements and inform the construction of the digital twin when those things depend on so many different elements in the digital twin. And that's the bit that I struggle with in this conceptually.
I don't think you can, I mean, you're never going to be dealing with just one measurement for a meaningful interaction with a multi-scale virtual human model. You're never going to be dealing with just one measurement, and you always want to perturb as much as possible. So you want to be making as many measurements as you can of function under all sorts of perturbations, exercise, etc., changing the environment. So I think it's much more than just taking one measurement and plugging it in. You're quite right. There's no way that you're not going to be able to plug that in as a boundary condition very easily. You've got to understand a lot of the other effects that are operating with it, you know, via the model for that control of that particular variable. I mean, I think a lot of the focus initially from a multi-scale perspective has to be about understanding homeostasis. To be, how does the body achieve homeostasis, and then of course adapt to changes, but how does it achieve homeostasis of a lot of blood biomarkers, including of course blood pressure and temperature, etc., etc. So there's lots of challenges. I mean, in the end, you've got to believe that if we're going to make full use of multiple measurements for an individual from multiple scales, dealing with the physics across those scales is the only way we're going to really be able to take maximum advantage of those measurements.
Yeah, the little bit of modelling I've done, I agree. It's all been based, the governing equations all been based on basically conservation of mass or conservation of whatever. Those are the things almost that you're able to construct your model from. But I'm still, what you're talking about there is construction of an integrated human model, trying to look at the homeostasis, things like that. That's fine. It's some sort of average model. And what I'm curious about is how you then take that step of actually having what was described there as the digital twin, the twin of me. Now, I can see things you could do. You could stick me in an NMR machine or a CT scan, and you can get the scaling right of the various organs. And I can see how to do certain things. I can see how you could do something that's quite big in terms of genomics and so on, where you could get the right isoforms of this and that here where they matter. You can sort of see how those things work in principle. So the bit I really struggle with is how, if you take, well, another example would be an oscillometry measurements on the airway, you know, how are you actually going to start working those in and start moving away from the generic human to the real twin?
I think we're hearing a real call there, Peter, for us as a community, Bioreme, who are actually the domain experts within respiratory, to work with the likes of Liesbet and Peter. And I just wondered if, you know, I'm just thinking about time really for the webinar, whether Liesbet and Peter, you can offer us as a network now some credible next steps so that we can do exactly what Peter has suggested. We have an enormous wealth of data, both modeling, clinical, immunology, and ideas, where we are writing grants and all sorts of things around modeling and multi-scale modeling but we don't really know what the international infrastructure is to support this. And I just wondered whether you can both close on some ways that we can integrate our initiatives within the Bioream network, whether that's organ level modeling of physiology to understand small airways disease, as Peter alluded to, or understanding how we can link single cell data in tissue to precision immunology within clinical trials. How can we work with you to get what we do into your frameworks? What can we do? Liesbet and Peter, maybe you could give us a few pieces of parting advice that we can then take back and come back to you with.
I think at this point for the EDIT platform, that's part of the whole reason of building such a VHD infrastructure, to find a place to communicate this, because there are nice initiatives also, for instance, in the anesthesiology community in Germany, where a few clinicians have consented to doing a certain level of annotation, and in exchange their data was pooled and was made available to researchers, and then they have nice data to work on, and in exchange the people that collected the data will get publications out of it, then they can get to use the results of the modeling. And these are small scale initiatives, but it's not always that this loop is so local or so easy to make. And that is exactly what the VHT is trying to do, find a way to put your information, make it available in the catalog so it can be findable, and you can find actually people that are looking for that data or for that expertise, so that these loops can be closed on different levels and for different organ systems. And at this point in building the roadmap, I think it's important to show that there is a request indeed from all sides. It's not just a tech push, but there is actually a clinical pool for these technologies. And so I invite everyone to either come to or send somebody to Amsterdam to have a look at some parts of the roadmap, and I'm happy to suggest which specific parts, or just put your thoughts on paper, communicate them, and I will make sure that they're in there so that these views are represented. And speaking now from the VPH Institute, we are more than happy to always connect people to the right researchers, or to help disseminate technologies across different organ systems, so that people don't have to reinvent the wheel when it comes to how to include specific measurements that are maybe multifactorial, and use them to optimize personalized models.
So I've taken away the Amsterdam meeting on the 15th, 16th of July, and maybe we will get back to you as a network with some thoughts on who might be able to come. But also, I think we have many clinicians across our network that might very much want to fill out your survey based upon the research that they're doing here in the UK. And Peter, you're going to have the final word, so that's okay.
Well, yeah, I mean, I think that there is a relatively sensible strategy here in terms of respiratory work, and that is via Merrin Tuffi. I mean, Merrin is already working closely with respiratory clinicians in Sheffield, a lot of contact with Melbourne, with some of the hospitals in Melbourne, as well as locally, of course, in New Zealand, and with various groups in the US. So in terms of respiratory, and she's very much plugged into this multi-scale framework that I've been talking about. So I would suggest, talking with Merrin about that specific link in relation to respiratory medicine.
Thanks, Peter. And I think Merrin is one of the international advisors for our network. So I think we will very much obviously continue to build that relationship. So I'd like to just end there, because I know we've run over time, but I'd really like to thank Peter and Liesbet for giving us some fantastic talks and an insight into both their research and the network more broadly. Thank you very much, and wish you all a really great day.